This is Dr. Sonia Shetty from the Department of Microbiology, and today we'll learn about what is the Ames test. Now you may have heard about the Ames test, which is a very common test used for detection of environmental pathogens. Let us see what it is and where is it used. First of all, there are many environmental mutagens around us. These mutagens or these chemicals that are around us, they could be natural, that they could be there in the food that we consume, or they could be even man-made. Many of these chemicals could be potential mutagens. Now, what are these chemicals? Where can you encounter them? They may be there in drugs, cosmetics, food additives, pesticides, and many industrial compounds like solvents. So, and we can be exposed to these kind of chemicals in a variety of ways. One is through ingesting food. For example, if you are having food additives which are containing these kind of chemicals, obviously you are going to ingest them. Second is through absorption through the skin. Some of them can get directly absorbed through the skin. And third, they may enter your body by inhalation. Now, many of these chemicals are or they can be mutagenic. So that is where we are coming to our topic, that is how can we detect these mutagens. Now first let us see what is a mutation. Now a mutation is a permanent inheritable change in the genetic sequence of an organism. Now mutations may not be desirable because it is changing your original genetic makeup, the original genetic makeup, that is the wild type genetic makeup or the wild type gene. So many of these chemicals that we talked about, they could cause mutations in your DNA which could be harmful. Mutagens can be physical agents like UV rays, they could be chemical agents, they could be even biological agents like some viruses. And many of these mutagens could also be carcinogens. So there is a difference between mutagens and carcinogens. Mutagens are those agents which can cause a mutation. Carcinogens are those agents or those mutagens which can cause cancer. So carcinogens are a class of mutagens. So they cause tumors in the body or in the tissues. So what is the Ames test? Now, normally, if you want to look for whether a chemical is a carcinogen, that agent or that chemical can be taken and it can be tested in animal tissues, and you can look whether it is causing any tumors. But animal testing is not an easy thing to do because obviously it is very expensive, it is time consuming, and so it may not be feasible to do animal testing for all for all chemicals routinely. So here we come to the Ames test, which was developed by Bruce Ames. And Ames test is a very simple and inexpensive method, a very fast method compared to tissue culture, but it is an indirect method, but it has a lot of practical applications. So what does this Ames test do? The Ames test assesses the ability of mutagens, that is these chemicals which are there in your environmental samples. It tests its ability to revert mutant strains of Salmonella typhimurium. So we have to take Salmonella typhimurium strains. So these Salmonella typhimurium, they are actually histidine auxotrophs. Now histidine, as you know, is an amino acid, and these particular strains of Salmonella, they are actually mutants which are unable to synthesize histidine. It means they are auxotrophic for histidine. That means that if the Salmonella has to grow in a medium, you have to provide histidine to it. So what do these chemical mutagens do? These mutagens can revert the histidine auxotrophs of Salmonella typhimurium. They can revert it back to histidine plus. We'll talk more about it in a minute. Now, Salmonella typhimurium is very useful. It is the tester culture use. So it is very useful because there are many strains available of this which can detect base pair substitution mutations and a variety of frame shift mutations also. reversion or a revertant. Now first, what is a forward mutation? So you have, as I said in the beginning, you have a wild type gene. When the wild type gene is mutated to a mutant gene, you call it as a forward mutation. Now that same forward mutation, if there's a second mutation at the same site, so that you can get a nearly wild type or a completely wild type phenotype back. It means initially there was one mutation, that is the forward mutation. The gene sequence where the first mutation happened, at the same place the second mutation happens, because of which it will go back to its original, that is wild type genotype and phenotype, or you can say a nearly wild type phenotype. So this mutation is called as a back mutation or a reverse mutation. So in this uh, test, in this method, we use a mutant strain of Salmonella typhimurium, and these mutants are auxotrophs for histidine. When this strain is exposed to a mutagen, it may revert back to its original wild type and it will now become histidine plus. That is what we have to detect over here. So these are the things that we require to do the Ames test. First, we require the histidine auxotrophic mutant. We require the histidine auxotrophic cells of Salmonella typhimurium. 10⁸ cells are taken. Second, we require S9 extract. Now, S9 extract is the liver extract obtained from rat, mouse, or hamster, these kind of animals. So, it's an extract from the liver. We will see why it is used. Third, we require a minimal medium which lacks histidine. Now, you have media, complete media like nutrient agar, which contain all kind of nutrients, allowing all types of organisms to grow. Minimal media will lack something because of which some particular type of organisms will not grow. Now, in this case, we are taking a medium which lacks histidine. It means that histidine minus cells, histidine auxotrophic cells of Salmonella typhimurium will not grow on this plate because this plate does not supply it with histidine. So, Salmonella typhimurium auxotrophic mutants will not grow on this medium. And the fourth thing that we require is a filter paper disc which contains that particular chemical that you want to test. Any test chemical, environmental sample, anything that you want to test, that is taken in a filter paper disc. So this is how the experiment or the test is done. So you have a histidine minus strain, that is a histidine auxotroph of Salmonella typhimurium. That is mixed with the S9 liver extract here. This is the medium which lacks histidine. Now on this, you spread this culture, the Salmonella typhimurium, along with S9 extract on this plate. So it is swapped on the plate. Now, you take a filter paper disc which is dipped or which contains that chemical to be tested, and you place it in the center of this plate. Now what will happen? If your medium or if your chemical is a mutagen, if it is able to cause a mutation, it will revert the Salmonella typhimurium back to its wild type. If the Salmonella typhimurium goes back to the wild type, it means now it is able to make histidine. So it is histidine plus now. So therefore, on this plate, you can see these white dots which are the colonies. So the Salmonella culture has grown on a plate which does not contain histidine because it has got reverted back. On the other hand, this is the control plate. In the control plate, you have to put a filter paper disc without the chemical. Obviously, it's a control, so that chemical is not put in the disc. Just the disc with saline or some controlled solvent is put. Now you will see here that the Salmonella typhimurium obviously will not get reverted back because there is no chemical, there is no reversion happening. So therefore, you will see around this disc there are no colonies, no cells. The salmonella remains histidine minus and therefore it does not grow. So on this test plate, these colonies around the disc indicate that the chemical which we had taken in the filter paper disc is a mutagen and it is mutating the Salmonella typhimurium. Now, you may observe that on this plate there are a few colonies here and there which are not really next to the disc, but on the plate a very small number of histidine plus colonies are there. These are because of natural reversion. Sometimes spontaneously mutant cells can get reverted back. So these cells or these colonies are arising because of a spontaneous reversion. That does not indicate that this filter paper is containing the mutagen or the chemical is a mutagen. So we have to look for colonies around the filter paper disc. So very simply, you have to just take the culture, histidine minus tray, mix it with S9 extract, swap it on the plate, put your chemical on the plate, and look for any colonies growing on the minimal medium. Now we come to what is the use of this S9 extract? Why have we added liver extract from rat and mouse? So, there are some chemicals which are that we are surrounded by, which are mutagenic. So when you do the Ames test, you will directly get the colonies growing there, you will get the revertants. But there are many chemicals which become mutagenic only when they are metabolized by your body. That is, the original chemical will be non-mutagenic, but when it goes inside your body, they are metabolized by your system, by your own body, and then they become mutagenic. So the problem is that if you do the Ames test directly with this without S9 extract, you will not get a positive test, you will get a negative test. That is why the S9 extract is added, because liver contains all these metabolic enzymes. The liver enzymes in S9 extract will convert the non-mutagenic to mutagenic, because this is what is going to happen in our bodies. So that is the function of the S9 extract that is added. So any chemical which originally is non-mutagenic, it will be metabolized and then you will be able to check whether it is mutagenic or not. So then it becomes a foolproof test. Some examples of compounds who behave in this way. That is, they become mutagenic when they go inside your body. For example, benzpyrene. Now, benzpyrene is found in cigarette smoke, coal tar, automobile exhaust. So, we are exposed to it all around us. Now, benzpyrene, if you do the Ames test without S9 extract, you will find that it does not give a positive result. It will give a negative result. But if you do it with the S9 extract, it will be metabolized and it is converted to benzpyrene diol epoxide. And this chemical is not just a mutagen, but it's also a carcinogen. So it means that when it goes inside your body, it is able to cause cancers. There's another example of of a compound which behaves in a similar way. That is, it becomes mutagenic only after it is metabolized in the body. That is xylan. Now it is a fungicide which is used for agricultural purposes. Now, one of the limitations of the Ames test is that sometimes you may get false or negative results. For example, nitrobenzene, it's a very common chemical used in manufacture of polyurethane, and styrene is used in the manufacture of polystyrene. Both these are known carcinogens. However, in the Ames test, they give a negative result. So, if any sample is containing these two compounds, you will think that it is a negative test and it will pass the Ames test. So, for chemicals like this, you may have to do other tests like the tissue test or animal testing. So, advantages of the Ames test are very clear. We can see it's a simple test. Just you need the specific strain of Salmonella typhimurium and you need the minimal medium which does not contain histidine, and it can be done in any simple lab. Though it's an indirect method, but you can do large number of tests with using these resources. Second, it has been used so far to detect a vast number of environmental mutagens, and with few exceptions, most of the times you get the correct result. And thirdly, you can also do quantification of this method. Now, how can quantification be done? Now, when the filter paper disc is put on the plate, there are colonies growing around the filter paper. Then the colonies are growing in the form of almost a circle. So the diameter of that zone can be measured, and this way you can get a dose response curve. That is, one can put different concentrations of that chemical on the plate. The diameter, zone diameter can be measured, and a graph can be plotted. So you get a quantitative response. If the amount of chemical is more, then the zone diameter of the growth will be more, and if the amount is small, you will get a small zone diameter. So it can be modified slightly to get a dose response curve. So we saw that the Ames test is a very useful test, a simple test which can help one to detect mutagens and potential carcinogens in our environment and chemicals which we are exposed to regularly in our daily life.